I really want to go to Tamu to visit Xiaoning and rest of you guys, because virtual seminar is really not very optimal. I really want to interact with people, but nevertheless the data variance still in the between of us. Hopefully the virtual seminar is smooth, and feel free to ask any question. I really want to communicate with you guys rather than just one-way interaction. Today my talk is translation of bioinformatics, go deep and go broad. This is the major title in order to attract you here. And the subtitle is working example in deciphering molecular heterogeneity of ovarian cancer. I will just use one disease, like my focus disease research as an example to go through my view of translation of bioinformatics. I'm not sure, I asked Xiaoning this question, what's the general background of the audience? If it's most of us from different data science perspective, or do I need to repeat bioinformatics is? Xiaoning, you let me know, or otherwise I can go ahead. I think bioinformatics is specialized data science domain, in my view, highly interdisciplinary, because we develop a method and tools, but there's a buyer, on top of informatics, there's a buyer. We're trying to understand the biology system, and we're trying to address the biology questions. So whatever bioinformatics tools could be coming from statistical domain, coming from computer science, that's not really restrictive. These are the very general term. A little bit of self-introduction, I'm coming from both aspects. I'm doing the methodology research. I feel the omics data, if you're familiar with the genomic, transcriptomics, epigenomics, what omics data really provide a tremendous opportunity for us to develop a novel methodology, computation methodology. On the other hand, it's also very critical we land on a particular disease or biology question so we can iterate, because there will be some particular insight and a challenge from the disease domain. So that's in general my research. I probably have three aspects of research. One is disease heterogeneity. The second, complex, genomics or transcriptomic variant. The third is clinical omics. And today, I hope I can spend most of the time to share with you guys, the audience, about my working example towards addressing some of disease heterogeneity question. First of all, why we choose this topic, why to study disease heterogeneity? Because every human disease, regardless is cancer or common disease, diabetes or neurodegenerative disease, they are all heterogeneous. Disease onset could be young or old, and the disease progress could be fast or slow, and their response to therapy, they could be all diverse. So definitely we have clinical goals to study the heterogeneity or the aspect of the disease. On the other hand, from data science or bioinformatics aspect, we really want to provide insight. DNA as a blue point, and the messenger and the proteomics as a dynamic profile of the cellular system. Can we learn anything from system? And go even bigger picture to the cell tissue level, whether we can appreciate the microenvironment, how the disease coming from a single cell and spread out to the organ, etc. So that provides a very unique, I would call, applied methodology domain for bioinformatics, which is subtyping. So subtyping meaning you can separate the same disease type into several subtype or subgroup of the disease. And I told Xiaoning probably I won't put any formula, but I'm still trying to put some high-level technique explanation what subtyping is doing. Basically, I think it's nothing fancier than doing the clustering initially. Think about it, you have matrix in, you have molecular feature, for example, 20,000 genes messenger or protein expression level, you have patient sample. Then you really want to, in the middle, you want to separate the patient into different patient group. That's we call subtypes. And meanwhile, another critical interest we have is to also review which feature they separate or predict which disease subtype, because then you get a bunch of the gene or moleculars that you can get back to the lab or basic biologies to understand their mechanism driving the formation of each subtype. But technology-wise, I feel it's pretty traditionally. It's like you're working on the observed data X, you're doing the regular clustering, or you can form as a latent space or latent variable analysis, working on the linear or the kernel space, or you can represent as distribution or probabilities model. And the subtype solution could be highly probabilistic. And you can also use other alternative, I don't know, computer science or graphical model to represent the patient or the feature differently. But you know, but why challenge? You know, why we're doing that? Because generally, we always have cursive dimensionality. When I mention, you know, if we measure the messenger RNA activity of 20,000 genes across 50 patients, there'll be, you know, 20,000 features and 50 samples. Or if I study methylation in some of my working example, we may have 500,000 features across a few hundred patients. So the way we form the subtyping or clustering solution could be highly arbitrary. And how do we know? Because unsupervised, we don't have guidance. How do we know we are overfitting? If I give you a noise data point or noisy like a data distribution, you still can generate a clustering solution or subtyping solution, but how do we know it's real? And how many subtypes? It's like a model order selection. And in a given disease, are these subtypes exclusive to each other, or they are gradient? Like one subtype, they can gradually from molecular perspective or disease perspective progress to the other. And sometimes I always ask, you know, how do you generalize subtyping solution? Because when we talk about machine learning, you have classifier. Classifier can be represented as something like support vector or regression coefficient, a set of coefficient. But how do you generalize the subtyping solution? On the other hand, because we're working on the disease model, we are not interested in any random combination of subtype. We're probably interested in one aspect of, you know, data point or the feature point. Then how do you carefully introduce the semi-supervision information into the subtyping learning? Okay, so let's start with ovarian cancer as a simple or focus example. It's not simple, just focus. Ovarian cancer is one of the leading very lethal, meaning like cause a lot of patient death disease. By large, right now the diagnosis is according to the pathology pattern. So there are like 4 or 5 pathology type according to the cellular and the tissue texture. One is called serous, the other mucinous, clear cell, and metroid. The therapeutic options are pretty limited. It's challenging as other cancer. And today we will zoom in even further. Sorry, did I have any? Okay. Allow me to even zoom in further just to one pathology subtype of ovarian cancer. It's called a high-grade serous ovarian cancer. In 95% of the diagnosis serous cancer, once the patient are diagnosed, most of them are senior women and very often will be already advanced stage disease, meaning their cancer already spread out beyond the ovary organ, fallopian organ to, like, for example, omental and sometimes liver, sometimes to the upper abdominal disease. So make it very difficult to cure the cancer or even treat the cancer. These probably set you a very basic tone of the clinical like aspect of ovarian cancer. And very quickly, like I just want to show you one picture. And here it's the earliest probably molecular subtyping work of high-grade serous ovarian cancer. From now on, I will always use the like abbreviation HGSOC, stand for high-grade serous ovarian cancer. At that time, people only use the customized microarray, measure, you know, a handful of hundreds or thousands of genes as a row, and each column will be individual patient. At that time, they find, okay, there are like 2 subtypes in a limited number of patient cases, only 36. And then moving to 2008, there's an Australia study. They enriched like almost 200 ovarian cancer cases. They did transcriptome profiling. They say, okay, there are 6 subtypes, regardless what they are. And this is a big consortium study, the Cancer Genome Atlas, as Xiaoning alluded to, funded by National Cancer Institute. So that's very systematic work. We measure like 500 plus high-grade serous ovarian cancers, mesothelioma subtyping. And then we find like there are 4 subtypes. The case become bigger. But I'm not sure you're familiar with the survival plot on the left, but basically this is just to show you on the x-axis is the number of years each patient is survive. On the y-axis is the probability. And they color by different molecular subtypes just to show you whether they have the survival, like a prognosis clinical outcome difference in terms of like a different group of ovarian molecular subtype. And no statistically significant difference. And when we look at these results in Mayo Clinical, we feel maybe TCGA as a combination of like 20 plus hospital and institution, their clinical follow-up data may not be so clean, and there's other issue. So this is my first author like paper, co-first author paper published right now already 7 years ago. Yeah, this is how I started my journey. We reproduce the subtype. This is still, we believe there are 4 subtypes of ovarian cancer, and we significantly separate their clinical outcome in Mayo Clinical cohort. Although the number of cases is actually far smaller than TCGA like a cohort. They have 500, we only have less than 200. But allow me to highlight some of the clinical outcome here. The purple line is the so-called mesenchyma subtype of ovarian tumor. If you pay attention to this median line, like these patients, they, on average, or take a median, they live shorter than 5 years. And this is like the longest surviving group of patients. If I still take, this is the median, like 50% of a chance, then probably they are around 5 years. So the difference is easily like 2 to 3 years survival difference. That was the early time, how we get interest in subtype, particularly in ovarian cancer, because such tremendous survival difference among patients. And there's some biology interpretation, what each subtype is doing. I probably, for audience purpose, I probably will just skip that and just focus on the data aspect. But basically, for the worst performing outcome group, mesenchyma, we feel because fibroblast cell in this tumor, they feed maybe the cytokine, maybe the nutrition for the tumor. And the immunoreactive subtype of the tumor, they tend to have the immune cell to help to control and kill the tumor cell. That's the intuitive biological understanding we iterated for years. And these molecular subtypes association with clinical survival outcome, it's not confounded or remain significant, even if we adjust for the other clinical covariate. For example, because we know like older patients, they tend to do poorly, or like the patient cancer stage become higher than they will do poorly. But even if we control for the remaining other clinical variate, the subtype very significant. And then this is the work I collaborate with other computational group. Then I began to ask a question, because that was the year of 2016, saying, okay, if you look retrospectively, we already have a lot of samples. Can we just put the samples together and ask across the board, how many subtyping solutions are stable across different like studies? And we conclude, okay, they said like this is the chessboard study. They're trying to form the different solution and cross-check each subtype solution across different cohort. They said there are 3 stable subtype, and the sample size right now it's like 1,300. And then right after that study, I was curious, can we unify all the available public ovarian tumor expression samples, like do a data science like study if nowadays you are cool. And this is question number 1 is if you browse through or integrate all the data, are there some like shared subtype? And the second question is more my clinical interest, like whether this like a discovery works, we always find a tumor or disease are heterogeneous, but come to the clinical perspective, whether some solution are meaningful. like can provide different clinical guidance or different hint that physician or surgeon can treat like a patient differently. That was the motivation. And this flow chart give you very high level view. You know, while we were doing this initial like a pooling study, I forgot like how many study. We gathered 14 public gene expression study, adding together the 2,400 ovarian tumor gene expression samples. More or less, we find their shared genes, like 7,000 genes. So we did a combining like analysis. Certainly, there's some technical details how we correct the study to study difference to make them comparable and could be interoperably analyzed together. And to give you like a high level view, after that analysis, we find indeed, okay, the molecular subtypes in high-grade serous ovarian cancer are highly reproducible. These two are highly condensed figure. On the top, the heat map, each row is just one gene, 1 out of 100 genes. We feel these 100 genes together, they can form unique feature to tell the subtype. And each column, it's one individual patient's tumor. On the top is more than 2,000 public day-cells tumor, and we use very like a dense color bar, like if you can see my mouse cursor, we have 14 studies. So these very colorful rainbow-like color indicates where the sample coming from each individual study. Overall, we don't see the major batch effect, and then we further verify the same subtype signature in our ovarian cancer Mayo Clinical, our own institution cohort. And we pretty much, validate the same conclusion. There are like 4 or 5 subtypes, and 3 of them are very consistent. Mesenchymal, immunoreactive, and proliferative. Regardless, what study or what like a subtyping system, you always find it's reproducible. And I think probably around the same time, TCGA, they also published the so-called proteogenomics characterization. This is a proteomics study. In protein, because the messenger RNA is just intermediate product, you have DNA as blue point and the messenger RNA carry the messenger, so you can synthesize to the protein. And in this proteomics study, they also pretty much share the same subtyping finding, where again, each row will be a feature by the protein expression, and each column will be individual tumor, where you can see the mesenchymal subtype, proliferative subtype, differentiated subtype. They are pretty stable and variable from proteomics aspect. With that, yeah, and by the way, like last year we published another even bigger study. I listed as 2019, that's when we submitted study. But we published another huge consortium study, more than 20 hospitals participated. There are 4,000 ovarian tumor cases measured for molecular subtype defining gene and prognostic relevant gene. But again, we validate that's pretty much 4 subtypes. And then for the bioinformatics perspective, as I indicated very early on, we're trying to solve the biology question. What's the mechanism? What drives it? And whether that provides a clinical opportunity or biology discovery opportunity, we can do something and target the disease differently. So yeah, certainly we use a lot of computational work through the process, but I will just skip through that and just give you a few highlights of the clinical research, the clinical relevant question. That's why we find like 4 molecular subtypes of ovarian cancers and why they are even matter, like, first, as I told you before, in our 2014 papers, mesenchymal subtype is the worst, outcome group. And 3 years later, we double amount of sample size we had and restrict all the patient to advanced stage, meaning their disease already spread out and the metastasis beyond ovarian organ. We still find the same trend and pretty robust. Mesenchymal subtype is significant with survival. And we also began to collaborate with surgeon because they are the first frontier. Once the tumor are so large, surgeon need to take the majority of the tumor tissue out. For ovarian, that's a painful and lengthy process. The surgery operation sometimes take between like 4 to 8 hours. We find mesenchymal subtype tumor, they have the lowest RD0 rate. Allow me to explain here. It's a little bit medical jargon. RD0 means after 8 hours surgery. Think about that tremendous labor-intensive works by a group of surgeons. Whether you can remove all the tumor tissue that invisible to your eye, the RD0 stands for residual disease equal to 0 centimeter. So if the patient, like all their tumors can be removed, at least visually, these patients have the best outcome because you leave little left over and the tumor clone to grow to metastasize, etc. And I will skip like a medical, like a jargon, but to be very brief, we find mesenchymal subtype, they have the lowest RD0 rate, meaning like they are the most difficult tumor to be removed by surgeon. That was very interesting. And then we have quite a few follow-up like a clinical study with surgeon as well. And we further find out not only mesenchymal is worse with survival and the hardest to surgically debug, they tend to go further. The mesenchymal subtype tumor, like the patient, they tend to have upper abdominal or miliary disease. Again, a little bit medical jargon again, allow me to simplify here. Upper abdominal because ovarian is in the like a bottom part of the body, like a woman's body. But the disease, if they spread further to very up and even, I don't know, reach to the heart, close to heart, then become very infeasible for surgeon to remove the tumor because that may kill patient. And miliary disease, meaning these diseases are small nodule here and there everywhere, like they spread. It's very difficult to manually one by one clean them up. Do we have a question? Okay. Okay, good. Actually, I'm not used to that. You can directly unmute yourself, ask your question. Ovarian tumor, yeah, it's pretty much symptomatic. They have a CA125 as markers, serum marker, and there's like a very long longitudinal study. I think in UK, British, you can use ultrasound or whatever, but there's no effective way to do the early detection. The challenge is ovarian cancer compared to the breast cancer, the incidence rate is pretty low. The breast cancer, you know, maybe is 1 to 5 or 1 to 10 women you have it. Ovarian cancer, like incidence is 1 to 100 or 1 to like 500, something like that. So from public health perspective, there's just no incentive to pre-screen so many cases with ultrasound and other things, where you have the consequence, you will induce additional false positive clinically and patient emotionally wise, you have to manage this. Yeah, so that's a good question, but that's basically one of the holy grail question of ovarian cancer research. How can we early detect that? Right now, we don't have good marker. Like CA125 is okay, but often it's a companion use when patient already have other symptoms, like a bowel obstruction or others. But thank you for the question. Yeah, I can see the message box. If you have any question, please just pop up. I will try to pose and address your question. Thank you. That makes me feel much better comparing to like virtual seminar alone. And the mesenchymal subtype also like associated with very high grade 3 and grade 4 complication. I swear to you, this is the last medical jargon I need to explain to you. Because the surgery process is so long after 8 hours, you can imagine patient will experience blood loss, right? And the multiple organ resected, dissected, removed. And therefore, the grade 3 and 4 complication in medical means like patient may come back to the hospital for another admission. Or grade 4, the patient may die within 30 or 60 days of the surgery. It's hard to say that, but sometimes we fear in that scenario, surgery may hurt patient rather than help a patient, because what's the point? You know, we hope we can, through the surgery, remove a majority of the tumor and help the patient to live longer or even cure the tumor. But in the end, we find out the mesenchymal tumor is such a nasty tumor. It associates all kinds of surgery and very bad survival and surgical outcome. And besides the mesenchymal subtype, we also study with like other subtype association with other therapy. And this is one of the clinical trial study retrospectively. It's called ICON-7. We experiment the molecular subtype versus ICON-7 Avastin, the treatment. And this treatment is super costly for patient. It costs like $100,000 per year, easily. And we find out there's only one subtype. They could show the benefit in term progression-free survival. So like a subtype may not only serve as a biomarker for surgery decision, but may also serve as a companion biomarker for clinical trial treatment response purpose. So like this like the third part of my presentation is just give you very quick maybe examples, because each of the example actually will have multiple publications associated with that how subtype can influence clinical, although maybe not clinical right now, clinical practice now, but can bring in different clinical perspective. So there's more research, more evidence can be accumulated. That's part of my interest too. That can make like omics or data-driven study to benefit the patient. But let's go back to a little bit like maybe the basic scientific question. We see these different subtype of ovarian cancer tumors, like survival-wise or surgery-wise or response-wise, they're different. But why they are different? Then we just cannot like stop asking, like what's a molecular mechanism? What's a pathway mechanism? What the cellular mechanism makes them different subtype? So this is in my original like 2017 clinical cancer research paper. Just use some routine maybe pathway or network analysis. We have some speculation. Without getting you lost or dizzy, I probably just focus the line of mesenchymal subtype. Where's my like a cursor? Okay, it's here. Mesenchymal subtype from molecular wise, we pretty much know they express like fibroblast genes, maybe cancer-associated fibroblasts. And pathway-wise, they have a lot of cell-to-cell interaction, like extracellular matrix, receptor interaction, and several typical crosstalk pathway. When we say the crosstalk is a tumor, it's not isolated. Tumor need to communicate with a nearby vessel cell to suck in the nutrition, need to communicate with the macrophage, fibroblast in order to invade and disrupt and get into the next organ. So mesenchymal subtype reflect a lot of interaction pathway and molecular changes and activities. And in order to appreciate that, I collaborate with other biologists just to design the, I'd say immunohistochemistry study. For each of the mesenchymal subtype genes, we find from the subtyping study. These are the 6 genes. The reason is when we have these heat map, if you remember, just gene by patient, these metrics or data form often ignore the spatial heterogeneity. Even if we say like these are the 6 genes, they have higher abundance in mesenchymal subtype of tumor, patient tumor. We don't know where they express. Are they from tumor gene or they were from the nearby non-tumor cells? And this is the most at early years, like surprising and depressing result to me, because we find out all these mesenchymal genes are not from tumor. It's not the tumor cells express these genes. It's from the stromal cells. Then what we're looking at certainly still puzzle us. And then I began to work with pathology more, just trying to appreciate, okay, whether from pathology aspect, we can decipher the molecular subtype, because from this study, even this is just a 6 marker, we began to realize there's tremendous heterogeneity, not only from patient level, but also from where the gene expressed from tumor or non-tumor cell. And the mesenchymal subtype tumor, there's another critical characteristic is it has denser plasma features that the tumor will reconstruct nearby stromos. I'm not sure whether you can, maybe I can use this as an example to circulate a little bit the cursor. Wait a minute. Okay, so these are the tumor cluster cell, and the remaining like a less dense area. These are the stroma, meaning immune cell or fibroblast, like other non-tumor cells close to tumor cell. Mesenchyma subtype tumor, we find out it tend to cause the destruction, and these are the low grade of tumor stroma reaction. Like tumor is here, and stroma here. They have less interaction. Like a tumor is not trying to reconstruct and change the morphology of the stroma, like environment. And this is on the pathology aspect, like the mesenchyma type tumor is doing something differently. So even though the marker is expressed from stroma, but maybe the cause are still the tumor. On the other hand, we know that like all these cells, they secrete different protein and the cytokine. They use that signaling to notice each other. What should we do? For example, if a tumor has DNA damage, it will express the HLA type 1 protein and recruit the immune cell, and the immune cell will sequentially kill the tumor cell. So that's a cascading or interaction among the cells. And with that of knowledge, we began to work with animal lab, like this is stand for the patient-derived xenograft model. Briefly, we took out the ovarian tumor, like our surgeon, not me, and then these tumor were still warm and alive, can be injected into the mouse animal model. Once these mouse grow, like they take the tumor and tumor can expand, we find out pretty much these tumor in terms of treatment response and behavior very similar to the original human donor tumor, the original tumor you inject into the mouse. The good thing of the human and the mouse difference, like from bioinformatics perspective, when we have the sequencing rate, like RNA sequencing rate you're familiar with, it's a fragment of messenger RNA, we can map to the human genome, we can map to the mouse genome, and therefore we can appreciate like which genes expressed in tumor and which gene expressed by stroma. And from that point of view, we be able to name quite a few genes, like only upregulated in PDX or only upregulated in human donor tumor. And we can validate them for therapeutic consideration. It's really the awareness that from the beginning, we only treat like each patient tumor as a single data point to now, we know each patient tumor at least have 2 data points. One is tumor expression, the other is stroma expression. And in the recent like cell studies, and you may know like the technology advanced so much, and people can do the single cell, profiling. Then they can measure every single cell in ovarian context. This is Canada Vancouver study group's research finding. They pretty much recapitulate our mesenchyma or immunoreactive subtype of finding in the single cell and spatial resolution. But to be high level, at a very high level, their conclusion is mesenchyma subtype. If you remember, that's the worst outcome, difficult to debug, like a subtype of ovarian tumor. They tend to be stroma restricted, meaning like these blue circle are lymphocytes, like immune cell. The immune cell will be excluded from the tumor. They cannot get into the tumor. They cannot kill tumor. On the other hand, if you still recall in my earlier plot, the so-called immunoreactive subtype of ovarian tumor always have the best survival outcome, because according to the single cell and the spatial analysis, they have the immune cell not only in the stroma, but they also can get into, they call it infiltrating into the tumor and to killing the tumor. And with that, certainly drag me to another, different entire world, the digital pathology. I have many ongoing works towards that direction, but without getting audience to the loss, I just give you an overview. Just continue my working example on the molecular or tumor stromal aspect. I get into digipathology and began to appreciate and categorize the cellular level, tissue level, and the cell-cell interaction level of ovarian tumor heterogeneity and map to subtype and therapeutic options. So you said like a 40, like a 240, right? Like,
yeah, definitely.
Okay.
10 minutes, yeah.
Yeah, but I'm pretty pretty close to the end now. I mean, with very quick like illustration, a working example, just using ovarian cancer and the different subtype as illustration, I want to take a few minutes to think about the data science perspective, Actually, there'll be like a growing opportunity for me to doing the data science on the omics and heterogeneous, omics approach. Because like a variety of reasons, like we have growing number of data modality and amount of data, DNA, epigenomic, what they saying. But allow me to simplify and to be specify, not really simplify, I think specify, what be the data science opportunity and the challenges for me, because I'm doing the subtyping for easily seven years now. Like I cannot keep doing the same thing, right? Like I have to ask a different question. I think one of the fundamental challenges is data source and generation difference. If you remember my box representation, each row is a feature and each column will be sample. In reality, when our study becomes so big, like across 20 hospitals, you have to realize there's collection bias. There's some systematic bias of clinical covariate or technical covariate. And even there's more advanced challenges. How do you collaborate without data sharing, right? Like, because there's different patient consent and the privacy concern. So these are the challenges coming from sample dimension. And from feature dimension, unfortunately, these data sets are not always friendly and ready for, for the data science methodology guy, because even we talk about messenger RNA alone, due to the historical reason, they were generated by different measurement platform. They may have different number of features, like 8,000 by micro and RNA sequencing 20,000 or nanostring only 300 genes. So in a learning schema, how do you accommodate this variety heterogeneity? I have no question. I hope the audience can give me more clue, because of like a wider spectrum horizon of the data science position you guys in. The second question, the data modality, like a different data modality, because most of my talk, like most of my example give to you, it's just one data layer, is messenger RNA, transcriptome. But nowadays we have the DNA level, we have methylation level. By the way, we also have another, manuscript being published soon on a methylation-based subtyping. Then we will have proteomics and and the different modification proteomics, let's say phosphorylation and glycoproteomics. The question is, how do we combine these different data modality? I can get it like mathematically, you can concatenate all the matrix to be a super long one, or you can use a kernelized approach, combine the distance. But I always feel probably unsatisfying, because in the end, what we're trying to achieve is still the causality. We're trying to connect the dot across different layers, how the genomic alteration may drive the molecular difference, and in the end, turn to the proteomics pathway difference and lead to the phenotype or the disease subtype difference. So that's both opportunity and challenge again. So the last aspect of opportunity and challenge is multi-resolution data. If several years ago, I never imagined we will have such tremendous amount of data, because if I talk about patient, at most a few thousand patients, that's where the consortium level collaboration. But however, for ovarian patient, like every patient will have tons of pathology slides, because stage 3-4 disease, they spread everywhere. It could be from mental, from bowel, from liver. You have many tissue. Not mentioned to that, like on each tissue level, you have tremendous thousands and collectively millions number of cells. And these cells interact with each other, as I indicate from one of my subtyping study example. maybe tumor is not the culprit, maybe stromal, and nearby the microenvironment, nutrient will grow the tumor is the culprit, should be considered. From that point of view, I even leave as open because I just see that tremendous opportunity if we can reconcile this multi-resolution data still yet towards the disease heterogeneity understanding. With that, okay, yeah, I still have one more slide. Okay, I was thinking even though in my seminal title, I use ovarian cancer as just one working example. I want to room out further. In the field, in the translational or disease or clinical study field, there's many other diseases. Like on the left are ovarian cancer and breast cancer. On the right, because they're related to some of my collaboration with neurodegenerative disease, Parkinson disease, Alzheimer's disease. Probably from very high level, you can find the commonality. Traditionally, like from etiology study, you have clinical factor. And from traditional clinical, like pathology aspect, you have different pathology, maybe heterogeneity, variability, you already noticed that. But for us, for bioinformatics and translational bioinformatics, it's growing and explosively growing opportunity to characterize the molecular and genetic heterogeneity. Not all of them will correspond to the clinical or the pathology heterogeneity, but the level of the resolution and the mechanism discovery we can enable is tremendous. Okay, with that, this is the end of my talk. Any question or comment?